Hi, welcome. Today we're going to go over a couple of free response questions that you might see during this unit on chromosomes, mitosis, the cell cycle, and meiosis. And you may not need to go over all of them. You might be more comfortable with some in comparison to others. So you can use this table of contents to click on any of the bullet points, and that will take you to that point in the movie. Feel free to watch them all or only watch a handful of them, whatever works best or helps you the most. Good luck and enjoy. Hey guys, welcome to your test review. Today we are going to talk about how to ace free response questions on the test. So let's talk about first just some overall test strategies that you can use that will help you ace those free response questions. And the first one I cannot stress enough, and that is define, 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 and then define some more. Whenever you see terms in short answer questions, you should really state what those terms are. It doesn't hurt, and it's going to demonstrate your knowledge and mastery of a certain concept. So anytime you see a term in a short answer problem or question, go ahead and define it. The second thing that kind of goes hand in hand with that is that you're never going to be penalized for writing too much. And the more you write, the more you give an instructor an opportunity to find the things in that question that are directly, or in your answer, that are directly relating to that question stem. So write as much as you know. Try to relate it and try to make it into a concise answer, but write more rather than less. And then the last thing is to outline your answer. Come up with a way that you're going to lay out your answer that flows nicely, that directly relates back to the question stem, and that isn't disjointed. If it's disjointed, it's going to seem a little bit like verbal buckshot, and nobody wants to have that on a test answer sheet. You can also use rubrics if they're available for an answer, or you can take a look at the point values for particular questions, because point values give you some indication as to how many points an instructor is asking you to make for a given question. Those that have fewer point values associated with them will probably tend to need a little bit less in terms of their answer than those that have a higher point value. So use that as a kind of guide to set up the outline for your answer. And remember, demonstrating that you can master a free response question is going to give you A's in classes, which is going to lead to you graduating college and high school, which is going to lead to you becoming a mad scientist, which is going to lead to you earning a Nobel Prize, and then, hello, world domination. So please, please, please use these, practice these. It's going to help you. So let's go ahead and take a look at our first free response question. Okay, let's go ahead and take a look at the question here. The question is, compare the unique differences between mitotic metaphase, meiotic metaphase 1, and meiotic metaphase 2, and then we see a point value, 5 points. And those 5 points give us a great indication of how many statements an instructor would be looking for in an answer. But keep in mind, it's always better to write more than write less. One of the other things that we're going to keep in mind here is we have some terms that we should define to get our answer across, and to make sure that we get that we demonstrate mastery here of our concepts. And that is, we should demonstrate that we understand mitosis, we should demonstrate that we understand meiosis, and we should also demonstrate that we understand what metaphase is, because that is something that clearly these two processes have in common. So let's go ahead and define each of those. So let's start with mitosis. So mitosis is the division of the nucleus in a somatic cell. And remember, somatic cells are body cells. Meiosis is a process whereby gametes are produced. Metaphase is a step that is common to both, and is where spindle fibers align chromosomes in the middle or equator of a cell. And then we're going to stay, so we're going to transition here. However, metaphase has several differences between the three. And by the three, I mean mitosis, metaphase 1 of meiosis, and metaphase 2 of meiosis. So we've done a good job here of starting off. We have included our definitions so that we have demonstrated mastery of concepts, and we have definitely written a considerable amount here already, which is good. Always write more rather than write less. And we have segued the question nicely from what they have in common to how they will be different here. So now that we have this down, let's take a look at how they are different. So I did this by bringing in some videos that I think will be helpful here. So this first video is showing you metaphase of mitosis. So let's go ahead and write that up here so that we remember that this is mitosis. And if we look, we see that chromosomes have been aligned in the middle of the cell. The way that they are aligned is very important, and how many of them are aligned is very important, because that's one of the big things that's going to differ here from mitosis and then meiosis metaphase 1 and meiosis metaphase 2. So in mitosis, we have 46 chromosomes aligning single file down the middle. And we don't see 46 here because this is an animation and it would be hard to do this animation with 46, but if it were a human cell, because this is a diploid body cell, we would see 46 aligning single file. The other important thing here to note that this is happening in one cell. So that's important too. So 46 chromosomes aligning single file through the middle of one cell. So there we have mitosis. So now let's take a look at meiosis metaphase 1. So here we have the chromosomes, and if you'll notice, the chromosomes here look very different than the chromosomes over here. And that's because these chromosomes have already been joined into what we call a tetrad. And remember, tetrad is a pair of homologous chromosomes that have actually physically connected at their centromeres. And it is the tetrads, or the pairs, that are aligning through the middle of the cell here. So we see that here at the end of metaphase 1. So here we have 23 pairs of chromosomes, and we'll say as tetrads, A-L-I-N, aligning in the middle. And we will also see that this is similar and that it is happening in one cell. Now let's take a look at metaphase 2 of meiosis. And one of the very first things you'll notice here is that now we have this process of lining up chromosomes in the middle happening in two cells. But it looks very similar to mitosis in that you are seeing the chromosomes lining up single file. However, what you'll notice here is that the chromosome number has been reduced by half because we have haploid cells at the end of the first division of meiosis. So we have 2 chromosomes in this animation versus over here where we had 4. So if we were to translate that to a human haploid cell, we would have 23 chromosomes aligning single file. And this would be happening now in 2 cells. So at first glance, it looks pretty similar to mitosis, but there are 2 big differences between metaphase 2 of meiosis and mitosis. The number of chromosomes lining up in the middle and the number of cells that it is happening in. So we have 2 cells and 23 chromosomes in meiosis metaphase 2, and we have 46 chromosomes and 1 cell in mitosis. And then metaphase 1 is very different than both of them because we have chromosomes aligning up with their pairs. So you should be able to take those differences and put them together in some coherent sentences to finish answering the short answer question. And then you would have a bang up free response question answer. All right, here's our next question. Explain what crossing over is, when crossing over happens, and why it is important. Be sure to include any relevant structures created and the process that creates them. And we see that it is worth 5 points. So that tells us that we need to make 5 distinct statements. Also, we should take a look at things that we should probably define. So we definitely need to define crossing over, and as we define crossing over, we're going to be bringing up some structures that we will need to define as we move through the definition of the crossing over itself. In terms of outlining my answer, my question stem kind of gives me a good place to start in terms of my outline. It tells me to explain it, tell when it happens, and to define relevant structures, and to list importance. So the way that I'm going to kind of set it up is I'm going to start with telling when it happens. And after I do that, I'm going to go into the definition of crossing over, and that'll bring up the relevant structures created and the process that creates them. And then lastly, I'm going to end with importance. So before we kind of break those 3 bullets down, let's take a look at what crossing over is. Crossing over is a process that happens during meiosis, specifically in prophase 1 of meiosis. And what happens is the homologous chromosomes actually physically join together at their centromeres into a structure that's called a tetrad. Tetra means 4, and that's because they have 4 chromatids amongst them when they are connected. Those tetrads then swap pieces of their chromatid, which contains various genes, corresponding segments of the genes. What that does is that gives us new combinations of genes on the chromosomes that have never been seen before. So that is a pretty big importance in terms of genetic variation and genetic diversity. So let's talk through each of these big bullet points over here. First, when does it happen? So crossing over happens during meiosis, prophase 1. And for our definition, we're going to say homologous chromosomes physically join at their centromeres through a process called synapsis. This forms a tetrad. Each homolog then switches corresponding segments. So come back over here and look at the animation. So if we were looking at the purple being the mom's chromosome, or from the mom, and the green being from the dad, the paternal chromosome, what we see now is that we have this piece on the dad's chromosome, or sorry, the mom's chromosome that is from the dad, and vice versa. On the dad's chromosome here, we have a piece that is now from the mom. So now we have these 2 chromosomes that were completely different than what they started out being, and this gives us a new combination of genes. So now that we defined it and we listed our relevant structures, so we explained the tetrad, we explained how the tetrad happens through synapsis, and we explained what is crossing over, this switching of corresponding segments, we can now go on to the importance. So the importance of this is it creates new combinations of genes providing for genetic diversity, which is what sexual reproduction is all about. So we have gotten our when, we have gotten our definition and relevant structures and processes. We have our importance, and we have really solidly answered this free response question. Okay, let's take a look at the next question. The next question is, there are 3 distinct ways in which sexual reproduction increases genetic diversity in organisms. Please list these 3 avenues and briefly explain each. So before I even go into the three avenues of genetic diversity in sexual reproduction, I'm going to define sexual reproduction because that's an important term here that is in the question stem that we'll need to know and to explain before we move on to listing the avenues that gives us genetic diversity in organisms. So sexual reproduction is the production of new organisms by combining genetic information or genes from two individuals or two parents. So it's really important that you state that definition, and then we can go into those three avenues for genetic variation. So let's talk about those three. So in terms of laying out an outline here for my answer, I'm going to start with those three avenues. So those three avenues would be crossing over, independent assortment, and lastly, random fertilization. I'm not going to really go in detail into crossing over again since we just talked about that in a previous question. If you want to visit that part of the video again, you can click on crossing over and it'll take you back to that section. I'm just going to quickly go back over it, but if you want to see it in more depth, just click on crossing over. But crossing over is basically the switching here of different pieces of homologous chromosomes. So we have the maternal and the paternal chromosome. They join through a process of synapsis to form a tetrad, and then those tetrads or homologs switch corresponding pieces of their chromatids. That ends up giving us new combinations of genes on both of those chromosomes, which leads us to having different genetic diversity. So crossing over is an important way here that we have genetic diversity. The next way that we have genetic diversity is with independent assortment. And independent assortment we talked about in class is a lot like a genetic line dance. So we have homologous chromosomes lining up down the middle in metaphase 1 of meiosis. And because we have 23 pairs, there are many different ways that those chromosomes can align in the middle. So here we have, if blue is the maternal chromosomes, we have one on this side and one on this side and one over on the other side. But because we have 23 pairs of chromosomes, we can have, looking at this example, 8 different ways that those chromosomes can line up in the middle. Here we have all 3 of the maternal chromosomes on this side and all 3 of the paternal on this side. Here we have 2 of the maternal down here and 1 up here. So the way that we have these chromosomes lining up in metaphase 1 can impact how the genes end up in all of the cells that will result from this particular cell as it divides. So independent assortment is a really, really important way that we get some additional genetic variety. So independent assortment is how the chromosomes align during metaphase 1 of meiosis. The different combinations of alignment produce different combinations of genes in the cells that result. So, independent assortment, genetic line dance. Easy way to remember that. All right, and then the last way here is random fertilization. And random fertilization is basically the fact that it is completely random which sperm fertilizes which egg. It is basically a race. Whichever sperm gets there first is the one that will fertilize that egg, and it will block any of the other sperm from entering that egg. And it's a completely random act. So the three combined here, crossing over, independent assortment, and random fertilization, all three are ways that we get shuffling of genes in the resulting offspring and provide genetic diversity. All right, let's take a look at another question. Define asexual reproduction. Be sure to include one advantage and one disadvantage, at least two examples of types of asexual reproduction, and an organism that employs each method. So before we get into the rest of the question and outlining the question, let's start with what we need to define. So first thing, it tells us we need to define asexual reproduction. So let's do that first. So asexual reproduction is the creation of offspring from a single organism and then we'll put the offspring are clones. Of the parent. So, creation of offspring from a single organism, and the offspring are clones of the parent, so genetically identical to the parents. All right, so now that we have our definition out of the way, let's look at what else we need to include here. So we got our definition. Next, we need to look at advantages and disadvantages, and then we are going to talk about examples. So let's start, since we did definition already, let's talk about advantages and disadvantages. And there are many here, and we talked about these in class. So, for sexual reproduction, the single biggest advantage is that it increases genetic variability. It makes the genes shuffle, and we talked about the ways in which they do that through meiosis, and we get all of this genetic diversity and this genetic variation, which is what in turn helps organisms to adapt their environment, and it also helps to speed up the process of a population's gene pool changing, which is evolution. But it does have its disadvantages, and we talked about some of these also in class. We talked about the fact that it has a high energy cost. It takes a lot of energy to go out and find a mate, especially if the environment is not particularly suitable for that search. Courtship is timely as well. It takes a lot of time and resources. You have to have a certain level of fitness to do that. And usually, in terms of disadvantages, there's some kind of sacrifice in the fitness, the ability to survive and reproduce in one sex to the other. So one sex tends to usually have an advantage when we talk about sexual reproduction. So that can be a disadvantage too. For asexual reproduction, there are a couple here, and the fact that it saves energy. You don't have to go out and find a mate. And for organisms that live in crazy places, harsh places, extreme places, that is a real advantage. You don't have to worry about courtship, which is also timely and costly in terms of resources. And it usually means that there's a greater increase in fitness for the individuals. They have a better chance of surviving and reproducing because they only are depending on themselves. In terms of its disadvantages, the biggest one here is that it does not produce any genetic diversity. So if we have a population that is genetically identical and some kind of virus or other disease affects that population, it's going to affect all of them. There is no chance for any of those organisms, if they are genetically alike, to have any greater fitness for that particular problem. Because of that, it's often difficult to adapt to an environment if you don't have a lot of that variation, and it slows evolution down. The population, the genes in a population will not change as quickly because they're not being constantly shuffled with each reproductive event, and the only way that they're going to introduce any new possibilities of gene combinations is through mutation, so it'll be slower. So those are our advantages and our disadvantages. You can choose to write a couple of those down to practice your short answer. And then let's talk about some examples. The first example that we talked about in class is fission, binary fission. And binary fission is I'm going to write it up here, binary fission. And we'll put a definition, because if you state a term in a question, you should probably define it. And that is when a cell divides into two or more parts, and the regeneration of these parts creates new cells. So there we talked about two organisms that use binary fission, and the one that is shown here is a prokaryote, bacteria. And remember that bacteria have circular DNA, and that's an important thing to remember because that will be on the test. So their DNA is circular. It's not in a line. It's not linear like our chromosomes. They have a circular chromosome. So we copy that circular chromosome, and then that cell splits into two. It is a pretty simple process of asexual reproduction in one-celled organisms. The other organism that employs this method of reproduction, binary fission, is amoebas. So those are two examples of binary fission. So a cell dividing into two or more parts, and the regeneration of these parts creates new cells. So simple splitting into two of a single cell creates a clone cell. All right. Next, the second example that we talked about in class is budding. And budding is pretty cool. And we'll write the definition for budding. And budding is when a new organism develops from an outgrowth or bud on another one and separates when it is mature. We talked about how in class that would be like me starting to get this growth on my arm, and then this growth on my arm got a little bit bigger and a little bit bigger, and then it started developing hair and eyes, and then it started talking to me, and eventually it fell off and it was a mini me, and then grew into an exact replica or duplicate of me, which is very sci-fi and creepy. But the two organisms that we talked about in class that use this type of reproduction are hydras. We passed around a sample of the hydras, and yeast, which is a unicellular fungi, yeast and hydras. All right, so there's our budding, and then the last one that we talked about, which is really cool and crazy, is fragmentation. And fragmentation is when a multicellular organism splits into fragments and each of these fragments develops into a mature individual. And our example of this is a starfish. So this is actually a starfish's arm that has been broken off of the main body. And you can see this arm is starting to regenerate all of the different other arms that it is missing. And eventually they will get big enough that you would never be able to tell that this arm was around before these four. So fragmentation is a pretty cool asexual reproductive strategy. So there we have it. So we defined our asexual reproduction. We talked about advantages and disadvantages. We gave two examples of types of asexual reproduction, and I did more than that, but you guys can pick just two. And listing an organism, and I listed more, but you're going to just list one that employs each method, and we would get our full 6 points.